All right, so welcome everyone to our OpenSim webinar. My name is Jennifer Hicks. I'm the OpenSim R&D Manager and also the Associate Director of our National Center for Simulation and Rehab Research. I'll be serving as the moderator for today's webinar. I'm pleased to welcome today's presenter, who's Kat Steele. She's joining us from the University of Washington, and she'll be presenting on Synergy-based control in OpenSim. So next slide, please. So before we get started, a quick overview of the goals of our webinar series. So OpenSim is a freely available software application for visualizing musculoskeletal structures and simulating movements of humans and animals. So the first goal of our webinar series is to showcase the cutting-edge research that's being performed with OpenSim. OpenSim is also a growing community of users. So the second goal of our webinar series is to provide a platform so that members of the OpenSim community like you can communicate and collaborate. Next slide. Before we get started, a few quick reminders about the format of the webinar. We definitely want to have time to address your questions, but we'll do those at the end of the webinar using the text-based Q&A panel. If you have any technical issues during the webinar, you can consult the guide on our website or send a message to the host of the webinar. So with that, now I'd like to introduce our speaker. So Kat Steele is an assistant professor in mechanical engineering at the University of Washington. Her research is focused on integrating dynamic simulation, motion analysis, medical imaging, and device design to improve quality of life for individuals with neurologic disorders. She earned her BS in engineering from the Colorado School of Mines and her PhD in mechanical engineering from here at Stanford University. In addition to integrate engineering and medicine, she's worked extensively in hospitals such as the Cleveland Clinic, Lucille Packard Children's Hospital, and the Rehab Institute of Chicago. She has been awarded an NIH K-12 Career Development Award in Rehab Engineering and is also the recipient of an NSF Career Early Faculty Development Award. Kat is an incredibly active member of the OpenSim community. She completed her PhD with our group here at Stanford. She's an OpenSim Fellow, has helped and attended several OpenSim workshops, and she's also a winner of a pilot project award from the NCSRR. So with that, I will let you take it away, Kat.
Well, thank you, Jen. Can you still hear me okay?
Yes, you sound great.
Thank you for confirming. I'm really excited to be here virtually with you all today to discuss some of our recent work where we've been using OpenSim to examine the theories and implications of synergy-based control. Now, I wish I could see all of your hopefully smiling faces, but I look forward to your questions at the end and for future discussions at conferences and other places where our paths might cross. So today, this whole research that we've been pursuing is really motivated by a question that I think many of you share and think about deeply on a daily basis, of how do humans control movement? It really is an amazing process, the diversity and variability of tasks that we can complete on a daily basis with such ease, but also when things go wrong, it's amazing how very simple impairments in our system can really impair our ability to move and interact with our world. Now, in examining this question, I do believe that OpenSim and musculoskeletal modeling has a lot to offer as a powerful tool to help us understand and explore this space. We often talk about our OpenSim as this musculoskeletal modeling platform, which we all enjoy using on a regular basis. And lately, I've been seeing people start to add another prefix to this in terms of neuromusculoskeletal modeling. And I do believe that this is a strength in an area that we can leverage OpenSim to help us better understand how humans control movement. Now we'll see how many other prefixes people add to this. Maybe we should start a poll on what comes next, neurocardio-musculoskeletal modeling. Who knows how many prefixes we can add. But I really do believe that adding in this neural control component and using this as a tool to think deeply about how we control movement offers a lot of interesting pathways. One of the unique examples that we can do with musculoskeletal modeling is that we can actually specify and test different control strategies. which we then can compare to experimental measures such as electromyography to test and see, is this theory for how we are controlling movement actually agreeing with the activation and coordination patterns that we see in both unimpaired individuals and perhaps also individuals with impaired movement. And so this can give us a platform also to start to evaluate patient-specific deviations. For example, I work very deeply with individuals with cerebral palsy, and it's often hard to tell if muscle activity is actually contributing to someone's impaired movement or just a consequence of how they move. And so we can use this as a platform to help us probe and explore how different individuals are controlling movement. Now, traditionally in OpenSim and in other modeling platforms, optimization is a core piece of how we examine how muscles are coordinated and activated to perform a given movement. So whether you like to talk about the redundancy or the abundancy of muscles that we have to control the human body, we have many more actuators than degrees of freedom. And so traditionally we've used these optimizations, whether it's static optimization or computed muscle control in OpenSim, to help us understand and look at the coordination of the many actuators that we have to control our system. Now, our goal in this research has really been to extend some of these optimization techniques in OpenSim to give us a way to test additional control strategies. And in particular, our group has been interested in looking at the implications of synergy-based control in understanding movement. And so our goal has been to provide a tool that we could use for both synergy-based control, but also that can have wider applications in OpenSim for anyone interested in coupling or weighting the activity of given muscles for traditional static optimization. And so at the end, I'll also be describing the plugin that we've developed supporting both our synergy-based control research, but also hopefully that some of you can use in your own research for coupling or weighting activity during optimization. So now I'll return to our original question, which was how do humans control movement? And as I said, our underlying motivation for developing this plugin and in this research has been to use it as a tool to help us better understand the implications of one theory, that of synergy-based control. So synergies are often defined as coordinated patterns of muscle activity that flexibly combine to produce functional motor behaviors. And so this theory of synergies provides that we can identify these weighted groups of muscles, or this low-dimensional space, that are typically calculated from experimental EMG, which can describe a given individual's muscle activity during a task. So for example, during walking or balance control or reaching, prior research has demonstrated that unimpaired individuals, 4 to 6 of these synergies can describe typically over 90 or 95% of our muscle activity during these very complex tasks. And so humans tend to be operating in this low dimensional space in terms of how we are coordinating our muscles. And some theorize that this represents a simplified control strategy as opposed to having to activate each muscle individually, or that these synergies may provide basic building blocks for movement, which we flexibly combine to execute different tasks. So to look at this more graphically, you may think in this very simple case of muscle synergies. In this case, we have 2 synergies describing 5 different muscles. And so you can see that each muscle synergy has different weightings for each muscle. And so during a given activity, for example, maybe during walking or reaching, each of those synergies is activated with a given pattern over time. And if we take the sum product of these to get the total activation of each muscle, we can see that even in this simple example, that our 2 synergies can describe the activation of 5 muscles during a task quite well. And so this theory of synergies has been used extensively for understanding and looking at and quantifying coordination of movement in both unimpaired individuals for a wide variety of tasks. And why our research group really got interested in looking at synergies was in our quest to hopefully help understand and improve movement for individuals with cerebral palsy and other neuromuscular disorders. And so for individuals with cerebral palsy, one of our persistent challenges is that every brain injury is unique. Cerebral palsy is caused by an injury to the brain at or near the time of birth. And because each brain injury is unique, everyone's going to move slightly differently and coordinate their muscles in a slightly different way. And so we've really been looking for new ways that we can quantify patient-specific changes in neuromuscular control. And that led us to look at Synergies as a potential framework. We know that Synergies are altered in both cerebral palsy and stroke. A lot of the foundational work by Clark and Lena Ting and Jinsuk Rowe and many others and Vincent Chung have shown that, especially in stroke, whether it's in upper extremity or lower extremity actions, that the coordination of our muscles and these synergies are even more low dimensional than unimpaired individuals. So for while during walking it may take 4 to 6 synergies to describe muscle activity in unimpaired individuals, for individuals who've had a stroke, typically 2 to 3 synergies can describe the majority of their muscle activity. And we recently examined the same thing in individuals with cerebral palsy, and in a study of over 500 individuals, we also found that synergies were altered in cerebral palsy, and that these individuals also seem to be operating in this low dimensional space. Further, in both of these populations, it's been shown that how much an individual's synergies are altered is related to functional impairment, and also some of our recent work has shown that they are also predictive of treatment outcomes. Individuals who tend to have more synergies more similar to unimpaired individuals tend to have better improvements in gait and walking ability after a wide variety of treatments, from orthopedic surgery to selective dorsal rhizotomy. And so this has really been our motivation in pursuing Synergies and understanding what are the clinical implications of this low dimensional space, and how can we use this clinically to hopefully help us better understand and differentiate between individuals with cerebral palsy to customize and improve rehabilitation. And so that gets us to Synergies and simulation. As I mentioned at the beginning, we can use simulation as a tool to probe and explore and ask some of these what-if questions to help us understand the theory and the implications of both synergy-based control and altered synergies for individuals who've had a neurologic injury. And so today I'm just going to give a few examples of how synergies and simulation can be coupled together. And in particular, I'll be discussing how we've been using it within the OpenSim framework to hopefully let others employ synergy-based control in their research. So to begin with, I'm going to go over the tool and the implementation that we've made to enable synergy-based control in OpenSim. So if we start out by looking at the typical OpenSim workflow, we have a basic process where we start off with our data from the motion analysis lab and model. So often we'll use one of the generic models that come from the OpenSim community, which we can scale based upon motion analysis data to our given subject. Using marker data from the lab and force plate data, we can perform inverse kinematics to understand an individual's joint angles and motion pattern. You might perform RRA or some other residual reduction algorithm to make sure your system is dynamically consistent. And then at the end, we have a variety of different tools available that we use to estimate the muscle activity to reproduce the kinematics and kinetics of a given individual's motion. And so whether it's static optimization or computed muscle control are probably the two that are most commonly used to estimate muscle activity using OpenSim. And so in looking at synergy-based control in OpenSim, we were extending the capabilities of static optimization, which performs an optimization where it's minimizing the sum of activation. It's not just squared, it can be to any power, but extending the capability of static optimization in this workflow. And so if we're taking our traditional static optimization to implement a synergy-based control, we modified static optimization to allow weighted groups of muscles. So if we have synergies that we know we want to provide and give as our input to the system, basically a set of constraints saying these muscles must always be activated together in these weighted groups. That is going to be our input to the system, which is our synergy matrix, which is sometimes referred to W in the synergy literature, where we have a certain number of muscles in our system. So for this upper extremity model, we might have 30 muscles, and then you can specify a given set of synergies. Now the output of static optimization is typically our muscle activations. And in between here, we have to solve for that set of synergy activations, given these constraints of synergies on our system and a given task that we want to perform. What are the synergy activations that can produce the desired and specified action? So traditionally in OpenSim for static optimization, we minimize activation squared to identify the optimal, quote-unquote, optimal solution for a given action. For implementing synergy-based control, we made the simple change of going from minimizing activation squared to instead minimizing these synergy activations over the given task. And so out of synergy optimization, you'll both get these muscle activations as you would normally, which is the sum product of your specified synergy matrix. And then you'll also get the synergy activations over a given task. So another way to think about this is if we were specifying for synergy optimization in an identity matrix, where we have ones along the diagonal and zeros everywhere else, this would give us the exact same output between static optimization and synergy optimization. But in contrast, we could also apply or give any set of random synergies to our synergy optimization. And in this case, these different muscles would be constrained to activate together, and solving for synergy activation squared, we'll be able to solve for the set of muscle activations constrained to our input synergies. So that's a basic description of how we've implemented this synergy-based optimization in OpenSim. As you can see, it's a simple extension to static optimization. And now I'm going to go through an example of how synergies in simulation can help us to better understand how an individual might control movement, to help us understand some of the theories and potential implications of synergy-based control. So I'm going to start out with an example from another group where you might be asking yourself, well, can synergies even control movement? Perhaps you might say, if I give it a system a set of 6 synergies, can that even control the complex walk dynamics required for walking or other activities? And so outside of OpenSim, this was a question that Rick Neptune and Jessica Allen looked at extensively, where in Sim they actually applied synergies to a model of walking and demonstrated that yes, in fact, 6 synergies could control the dynamics of unimpaired gait, and that those synergies had relatively predictable actions helping to support and propel the body during this motion. Now in OpenSim, we were able to see that yes, synergies can control movement, and in implementing the synergy optimization, we had a slightly different question that we initially wanted to ask. We wanted to ask, well, can these algorithms that we're using to calculate synergies, can they actually identify an underlying known set of muscle activation or set of muscle weights in synergies? And so, for example, if I was to specify and give my model a set of synergies, have it execute a dynamic task, if I applied these methods that we're using to calculate synergies, can we actually take from those muscle activations and get back to those underlying known set of synergies? Or another way to look at this is traditionally we calculate synergies from EMG, where we have our muscle activity measured in the lab. We apply some type of matrix factorization algorithm. Non-negative matrix factorization is by far the most common, but we use that to decompose and identify these weighted groups of synergies that can describe the muscle activity during our motion. And so in this analysis, I'm going to share with you how we use this synergy optimization plugin to help us look at this question of, can we identify a specified set of synergies from a dynamic task in action? And how confident should we really be in these muscle weights? And so I'm just going to go over this briefly today as an example for how we've been using this plugin in OpenSim, but you can also find all the details of this analysis in our paper in the Journal of Neurophysiology. All right, so hopefully I haven't confused anyone too much so far. It's much more difficult when I can't see all of your faces. But we'll go ahead and now dive into a little bit more of the methods we use to look at this question regarding if algorithms can identify synergies. And so we basically started with a musculoskeletal model. We gave it and specified different sets of synergies. And from that model, we were able to calculate the activations predicted from our synergy optimization that both satisfied the dynamics of the task and were also constrained to that input set of synergies. We then wanted to apply a set of these matrix factorization algorithms, such as non-negative matrix factorization, to identify a set of synergies. And our underlying question was, how well would these synergies match? Could we basically back out the synergies that we knew were underlying and specified for the system based upon our muscle activations and these matrix factorization algorithms? So to test this, we used an experimental protocol that had been previously used for by Jinsuk Rowe and Zev Reimer for evaluating synergies and muscle coordination in both unimpaired individuals and individuals after stroke, where an individual grabs onto a manipulandum and without moving their arm generates forces in a variety of different directions. And so we recreated this experimental protocol in OpenSim, where we had our model positioned similar to the subjects in the study. And we had, now the subjects in the study, I forget, they either applied forces in 30 or 100 directions, but another great thing about models is that they never get tired. And so we had our model produce a 10 Newton force in 1,000 different randomly assigned directions around the hand. So that was our task. Now, for each we had the model execute the task with different sets of specified synergies. And similar to a prior study by Matthew Tresh, we specified these synergies by choosing from a random exponential distribution. And so we'd give the model a set of random synergies, have it execute the task, and we would get, with synergy optimization, then solving for those muscle activations, where we were minimizing those synergy activations C squared over the task. Once we had our muscle activations as our output, we then applied a wide variety of matrix factorization algorithms. As I mentioned earlier, non-negative matrix factorization is by far the most common used to calculate muscle synergies from electromyography, but we also tested a variety of others, such as principal component analysis, independent component analysis, and a couple of other variants of those too. And so from each of these matrix factorization algorithms, we were able to identify our set of synergies, and then we calculated the similarity of those identified synergies to our original specified synergies by calculating the average correlation coefficient. Now, in this case, we had our musculoskeletal model and the dynamics of the system, but we also wanted to test what the effects and our ability to identify synergies were without a model. So we also, in addition to having it execute the task, just had a case where we identified random coefficients, random C's, to get our muscle activations, and in that case also calculated our identified synergies and compared back to our specified synergies. And we repeated this for 100 different sets of randomly assigned synergies to see how well we could identify the underlying known neural control for our neuromusculoskeletal model. All right, so any predictions on what happened? We'll go ahead and see. Well, so I'm going to start out with the without the model effect. And so without the model, we had similar results to another study that had been earlier published by Matthew Tresh, where if we assigned random synergy activations, you can see that some algorithms actually did pretty well in identifying those underlying synergies that were specified. So non-negative matrix factorization, probabilistic independent component analysis had a normalized similarity greater than 0.7. And so here I'll note that this is normalized similarity, where we normalized it such that 0 was that similarity due to chance, because especially when we're comparing large sets of vectors, you can actually get pretty high correlations just by chance. So 0 is correlation due to chance, and 1 would be perfect similarity. So looking at this, and similar to Matt Tresh's earlier analyses, there were some algorithms that were able to identify the specified synergies pretty well. But again, this was without the model. So what happens now when we add in the model and these biomechanical and task constraints? Well, what we found was that with the model, the ability of all of these algorithms to accurately identify our synergies decreased significantly, except for PCA, which was pretty poor to begin with. But overall, we saw that the ability of these algorithms to identify the synergies decreased to less than an average of 0.5 in our normalized similarity. And so somehow by having just the biomechanical and task constraints specifying which synergies were being activated, we had a difference in the ability of these matrix factorization algorithms to accurately identify the synergies that we knew were constraining and underlying the system. So what's going on here? If we look at our system, we had our specified synergies, our synergy coefficients, which we were using to calculate our muscle activations, or what you might consider like our EMG in our model. And so without the model, our specified synergies were identical. We used the same ones across the board. The place where we had difference in our system was in how these synergy coefficients were calculated, that C matrix. Without the model, it was this random exponential distribution, just specifying do anything you want, no biomechanical or task constraints. While with the model, it still had to produce force in 1,000 different directions at the hand. And so how synergies were activated tended to be sometimes coupled slightly more. And so we knew that it was in the synergy coefficients that this decrease in the ability to identify synergies had to lie. And in our case, with the random exponential distribution, you can think of it as that the synergy activations were really independent. They were not correlated at all since they were random. Every synergy was independent. However, when we add in a biomechanical and task constraints to our system, some of the synergy activations start to get correlated. So an easy way to think about this is if I was trying to, for example, generate a force upward with my hand, and I had two synergies that maybe favored your deltoid muscles at your shoulder, those two synergies may often be activated together to execute the task. And so how the individual synergies were activated were no longer perfectly independent from one another. And so we wanted to investigate if this effect of correlated synergy activations can have a significant impact on our ability to identify underlying synergies. And so for that, we returned to our case without the model. And so here I'm just showing again for each one of the algorithms that normalized similarity without the model. And in our initial case, since it was a random exponential distribution, the correlation between how any two synergies were being activated was 0. They were all just random and independent. But we asked, well, what happens if this case without the model, we start adding in more correlation. So we start making it so that there's increasing correlation between how different synergies are activated. And what we saw was as we increased the correlation from 0.1 to 0.23, 0.5, 0.7, 0.9, especially for non-negative matrix factorization and any of our independent component analysis algorithms, that we did see this decrease in our ability to identify those underlying synergies as that correlation increases, which makes especially total sense when you're thinking about independent component analysis, because as soon as they start to be correlated, they're no longer independent. And similarly with non-negative matrix factorization, these underlying independent components that we're trying to identify, we see a decrease with increasing correlation. And you can also see that this compares, although it doesn't fully explain the load normalized similarity that we had with our biomechanical model. But it provides evidence that because of the biomechanical and task constraints that we're dealing with when we have human motion, that just by having actions that are correlated that require certain muscles to be activated together more frequently, we decrease our ability to accurately identify the underlying synergies. And so what does this mean for that original question that we asked? Can the algorithms identify synergies? Well, first of all, I will say that all these algorithms still could identify the low-dimensional space. So they still were able to identify, yes, you're operating in a low-dimensional space consisting of synergies. We know that this low-dimensional space exists. However, where we saw that they really struggled was to identify that orientation in the space. So I like to think about it if I was in a three-dimensional space, these algorithms can all identify that, hey, you're in this three-dimensional space, but how you're actually rotated within that space is actually really hard to identify. And that's the factor that is associated with these muscle weightings. We can easily identify for a given individual and in a given task that our complexity of control is low dimensional, but it's very hard to identify which set of muscles are activated together, and the weightings are highly dependent upon the methods and the matrix factorization algorithms that you employ. And that these are really highly tied to that correlation of synergy activations. Our abundancy of muscles lets us execute a wide variety of tasks, but it also means that we'll often use similar muscles to execute similar tasks. And so we have kind of this built-in correlation in our system that decreases the ability of these matrix factorization algorithms that often implicitly assume some type of independent underlying components from really working very well to describe our underlying motion. So what does that mean for synergy-based control? Should we just discard it and get rid of it? Well, as I said before, one of our main drivers in all of this was looking at, well, how clinically useful are these synergies? And for us, we still are seeing that synergies and a description of how individuals are coordinating their muscles and this low dimensional space is highly predictive of such things as treatment outcomes. And so they seem to be clinically useful for helping us to quantify and identify different individuals within, for example, the spectrum that is cerebral palsy. And then the results of this study, though, have led us to more focus on dimensionality. So, for example, saying that a given individual can describe 96% of the variance in their muscle activities with 3 synergies, as opposed to, for example, focusing too much on these individual muscle weightings, which we know are more dependent on the methodologies that we use. I also do think that it does provide a useful framework for still identifying these patient-specific models of altered control. If we can specify this low-dimensional space and identify how it's altered for a given individual, whether they have cerebral palsy, stroke, or some other impairment, we can still identify and describe this space to help constrain and understand the implications for altered movement. So when we're looking back here, we saw that with some of the great synergies and simulation work that we can really use simulation as a tool to help us understand both the theory and the implications of theories of synergy-based control. And so we saw from the work with Jessica Allen and Rick Neptune how we can look at questions such as, can synergies control movement? And in our case, we were looking at, can algorithms accurately identify synergies? And we saw that there is some caution there. These matrix factorization algorithms that we have some built-in assumptions that may not always perfectly apply to our human complex musculoskeletal system. And so we need to be aware of those assumptions, and especially as we continue to move these tools into the clinic to apply those methods appropriately to each individual. So that's kind of an brief overview of how we've been using this synergy optimization plugin to help us kind of dive in and explore some of the underlying meat of how synergy-based control can be useful for examining human movement. But I did want to close today by talking about some of the other applications for how this plugin may be useful for you or for others that you know in your research. So we originally developed this algorithm for synergy-based control to give us the ability to couple the activation and give weights to a bunch of different muscles within the system. However, the same framework also lets you do a variety of other things. For example, often if we want to, if we're in a simulation, we may want to give different muscles different weights to either penalize or favor different muscles when we're performing a given optimization. And so in that case, if instead of having an identity matrix here, you can imagine that if you replaced the element for specific muscles with a different weight, you're able to penalize or favor different muscles with respect to each other when you're solving, when you're estimating your muscle activity. An example of how we've used this in the past is back in 2011, we were looking at how we could estimate tibiofemoral contact force to examine how contact force changes for individuals with cerebral palsy who walk in a crouch gait. And in this study, we identified a set of muscle weights that provided the best match to experimental data. And so we had data from an instrumented total knee replacement, and we also had our estimated force from our OpenSim model. But since we know that our muscle activity around the knee is a big driver of these tibiofemoral contact forces, we looked at how weighting different muscles like the hamstring or the gastroc or the quadriceps had an impact on that estimated force and how that impacted our relation to those experimental TKR force. So in this original paper, we hard-coded those weights into the OpenSim source code, but you could apply this similar framework using the Synergy optimization plugin as well. Now, another potential application of this plugin is if you're interested in coupling muscle activations. So for example, maybe you look at that gluteus medius muscle in the OpenSim model and say, I'd really like for those three muscles to always activate together. I really don't believe that the anterior compartment can activate completely independently of the posterior compartment. Well, if you wanted to test that out and look at the effects, instead of providing an identity matrix, you could provide a matrix where now you have a different set of muscle groups, and by adding a column with ones for all three components of our gluteus medius, you can couple the activations of those together. So those are just some simple examples of how this plugin can be used for a variety of different tasks beyond just synergy-based control. And so it is all based on static optimization and builds upon the static optimization code. But I hope that you all will test it out, send questions. We like to see things continue to grow and hopefully become even better by your input and your uses. And so this is available on SimTK. You can see the website in the upper right of your screen, simtk.org/home/synergy. But I'll go through, first of all, just one quick example, and this is also the example that is provided when you go to the SimTK website. There's the plugin, a set of MATLAB code for creating setup files, and instructions for this very simple example, which uses the ARM26 model, which comes with OpenSim. And so if you were to open this example, you'd see the MATLAB code for creating the analysis setup file. But as I said before, this is pretty much identical to static optimization. And so if you open the XML of that setup file, you'll see that it's pretty much identical, except for under analyses, instead of seeing static optimization, you'll see that you need to put static optimization 2. I'm very creative in my naming. And one of the other differences that you'll see is that you do have to specify that what we're calling the activation weighting matrix, or that W matrix that I was showing you earlier. So in this very simple case of the arm, you can see that each vector has the number of muscles or number of actuators in our system. So in this case, this model only has 6 muscles. And so in this case, this activation weighting matrix is just an identity matrix and will give you the identical results between static optimization and this tool that we developed for synergy optimization. So look, I'm going to go through just a few quick examples that you can go through later also and try on yourself using this ARM example. So what if we say, well, first of all, let's actually compare it to static optimization. If I just take an identity matrix, here's this model, and you saw it before, the arm is basically just flexing the elbow. And here's the activation of our biceps femoris long head and biceps femoris short head predicted by static optimization to execute that action. And I will mention that the order of these muscles is important. So if you go into your model, you would see that the biceps long head and biceps short head are the 4th and 5th actuators in this model. And so these would be the weights that we'd want to be changing if we wanted to affect the activation of those muscles. So first of all, if we just do the identity matrix and we do static optimization and then synergy optimization, you see, as expected, that you would get the identical activation. Now, what if you came in and said, well, I don't know, perhaps you gave Botox to someone's biceps femoris long head, I have no idea, but you wanted to penalize the activation of that muscle, or maybe you had really fatigued that muscle, and you wanted to see what happens if I penalize that activation of that muscle. What other muscles will compensate? And so one way you could do that is by changing the weight of the biceps femoris long head to 0.1 to basically penalize its activation in static optimization. And so if I were to run with this activation weighting matrix, You can see that, as expected, the activation of the biceps long head decreases significantly, while that of the biceps short head increases to flex the elbow. One other thing you might want to do would be to group muscles. So say you wanted to make the biceps long head and biceps short head activate together. So now instead of having 6 vectors, you would only have 5 vectors, and in one of them you'd have ones for both the biceps long head and biceps short head. And in that simple case, you have to take my word for it, but there's 2 gray lines, one on top of the other right here. But as you can see, the muscles have the same activation level. And so you can use this simple example to hopefully play around with and get familiar with the basic format for how you can use this plugin, whether it's for synergy-based activation, weighting muscles, or coupling the activation of muscles. And as I said before, it's available on sympk.org under downloads. And so feel free to let us know if you have any questions or comments or other requests. So with that, I'm going to wrap up so that we leave some time for question, and wanted to say thank you to you all for taking the time out of your day to join in. I want to thank all of our members here at the Ability and Innovation Lab at the University of Washington, Eric Perot and Matt Tresh, who were the partners in crime for looking at the impact of different algorithms and biomechanical and task constraints on our ability to identify synergies, and also Mike Schwartz and Adam Rozumowski at Gillette Children's Specialty Healthcare, who are our partners in crime looking at how synergies are altered in cerebral palsy. I also want to give a special thanks to Ayman and the rest of the OpenSim team for supporting us in developing the plugin and answering questions throughout the process. Thank our funding for this project, and I will gladly take any questions that you have. So thank you all.
All right, thanks, Kat, for the great talk. Really interesting results and super clear, even though you couldn't see all of our faces. Now I'll go ahead and get started with the Q&A session. So as I mentioned at the beginning, we'll take all the questions via text. So you need to find the Q&A panel in your WebEx controls, type in your question, and make sure you select to ask all panelists. So while some questions are coming in, I'll go ahead and ask a question. So, Kat, how the synergies that you started with, were those randomly generated, or were those based on EMG?
For the study with the upper extremity model, they were just random synergies. So we, using a process similar to what Matt Tresh had done in an earlier 2009 paper, we drew those synergies from a random exponential distribution, which has been shown to be similar to the EMG data from some prior analyses, but they were all random synergies.
Okay, so did you examine like how much correlation? Like, I guess my higher level question is, do you think you would get better recovery of the synergies if you had better synergies to start with that better gave an independent mapping of the motion?
Yeah, and that's an interesting question. And part of our reason in doing the random was so that we were hopefully covering the space of potential and possible ones. And you probably saw that for some of the algorithms, there was even a pretty big variation. For some synergies, you might be able to have a normalized similarity of like 0.8, and for others, you'd be down at like 0.3 or 0. And so even for individual synergies, there was some variation. And so one thing that we did look at was the similarity of those random synergies to the original synergies that were from Jinsuk Rowe's paper. And so, of course, there are some limitations there because there were 8 muscles, I believe, with EMG. And so we had to look at kind of a lower subset of the muscles that were in our model. But we didn't find any significant correlations between, for example, like how similar are these to the ones that had previously been calculated from EMG. But granted, again, that those come from non-negative matrix factorization. So we didn't see any correlations there. I will say that one interesting other element, which is in one of our other papers, but using the same setup, is that if you take static optimization, just plain, and you calculate the activations, the resulting synergies that you get from those activations are also very similar to the EMG, to the synergies calculated from experimental EMG. So whatever this low-dimensional space seems to be describing, we're able to get it both through optimization frameworks and some of these matrix factorization algorithms. So it's a little bit of a chicken and the egg question, which came first, the synergies or the...
And then another question I had, and this one is a little tricky because you get better recovery of the synergies when you have no model than with a model. But have you tried to examine at all how the accuracy of the musculoskeletal model is related to the synergies you recover from those algorithms?
No, we have not looked at like how accurate our model is impacting the accuracy of the predicted synergies, but I wouldn't see that as having a huge effect, or I wouldn't see it as like making our ability to identify it better. I've tried a similar thing with a lower extremity model, and you get very similar results. It's an interesting question. I'd be curious to see what is the factor that you think if you had a more accurate model would lead to a more accurate ability to identify the underlying synergies.
Well, I guess your results that you presented would suggest the opposite would be true, since you get better recovery when you have no model. You know what I mean?
Well, that again comes back more to the fact that with no model, the activation of the synergies is totally uncorrelated. Well, once we have the model, since there's often muscles that'll do more similar actions, we tend to have synergies that tend to be more highly correlated. Just, synergies A and B are used more often when you're exerting a force up as opposed to down, while synergies C and D are exerted more often when you're making a force to the right. And so it's more tied to that underlying correlation because of the fact that we have an abundance of muscles that can be used for a task.
Right. Okay, we got lots of questions from the audience too, so I'll jump to some of those. So a question from Jeff Reinbold. So he says, very nice talk. How different can a movement task be to see a difference in synergies? And also, do you see synergy differences between genders?
I've not looked at the gender question, but easily could. I guess one interesting thing is that our kinematics, kinetics, and EMG, for example, for walking are very similar between men and women. So my guess would be very little, but we can look at it and get back to you. What was the first part of the question again?
How different can a movement task be to see a difference in synergies?
Oh yeah, and so that's an interesting one, and one that we're really interested in, because especially when we're looking at gait and cerebral palsy, we've been very curious about, do individuals with cerebral palsy have different synergies just because they walk differently? And so if I had you walk in a crouch gait or some other motion pattern, would you also have altered synergies? And so that's actually a question that we looked at recently as well in partnership with Marjolein van der Kroot at the VU in Amsterdam, who had previously had unimpaired individuals walk in a crouch gait and collected EMG. And so we presented this at a conference last summer, and the publication is under review. But we did see that when unimpaired individuals walked normally, and then when they walked in a mild and a moderate crouch gait, that synergies did not change significantly. And we've also looked in individuals with cerebral palsy to see if there's a correlation between the change in kinematics and the change in kinetics and how similar two individual synergies might be. And we also there see no correlation between similarity of kinematics and similarity of synergies. And so those both have kind of provided evidence for us that altered synergies are not just due to altered movement.
Okay, so now a question from Pablo Ortega Oriol. So what is your view on the manyfold or minimum use theory? That's the theory that includes synergies but aims for a reduction of dimensionality based on a performance variable. Would you consider this for future research?
Synergies are an example of, kind of this reductionist approach. And I think what's clear is that we're operating within a low-dimensional space. But what is less clear is what's driving that underlying space. Do we have underlying central pattern generators that are, basically like we're doing to the model, constraining certain muscles to have to activate together? Or has our body, for example, learned that these certain couplings of muscle activations are optimal for energy consumption or whatever optimization you prefer to look at? In our research moving forward, we really are most curious in clinical utility. And so we're focusing most of our time in looking at what are the clinical implications. But I agree that it's a very interesting area to think about what are these underlying synergies representing. And once again, getting back to that question of how do humans control movement and what are the really kind of fundamental building blocks that we use in daily life.
Okay, so now a question from Puneet Singh. So this is a question that I've seen come up before, and so he or she's interested in your viewpoint. Might synergies only be a mathematical construct coming due to the task?
Yes, I think, as I just mentioned, I think absolutely. So if you look at some of the animal work, so we have the animal work and the human work, and there's many people who could speak more deeply on this than I could. But in the animal work, for example, when they stimulate the spinal cord, or if they're able to do things that we fortunately can't do to humans, but there they do see that there is some coupling of muscle activations, which means that there probably are some pieces of the neuroanatomy that tie and couple muscle activity together. But there's also this question of, are synergies such as we're calculating with these matrix factorization algorithms, a mathematical effect? And I think that there is some evidence that, yes, we're really not sure, and it's once again this chicken and the egg problem, which came first? Are we optimizing and that happens to be in a low dimensional space, or is our neuroanatomy actually set up so that there's a low dimensional space that we have to use for a given task? I will say that whether they're a mathematical artifact or not, we are, especially in cerebral palsy, seeing that it's a very useful tool for characterizing and quantifying motor control. And so I think understanding what this low dimensional space is and how it might be useful for identifying patients is kind of the pathway that we're taking forward.
I'll also briefly share one study that's somewhat relevant, and one of the favorite studies that I've read recently. It's actually an old study from the 80s by Esther Thalen. And so infants have a stepping reflex when they're really little, but then it goes away. They don't know why. And so they took a bunch of babies, some who still had the stepping reflex and some who didn't. And the ones who didn't have the stepping reflex, they put them in water and they had the reflex again. And the ones who still had it, they put little weights on their legs and they couldn't do the stepping reflex anymore. So basically, babies just get too fat, and that's why they're not strong enough to lift up their little baby legs. So,
and I'm going to feel bad if I get this wrong, but I believe it's Dominici did a synergy study that was published in Science, where they actually calculated synergies from this infant stepping, and you see that there's these underlying patterns that are preserved both in humans, as they grow from infant to toddler to young child to adult, but also that she examined several other species as well and saw similar effects. And so there do seem to be some underlying pieces that drive it. And I will also say that when we look at synergies in individuals with cerebral palsy and stroke, that lower dimensional space that we get for these populations tends to look more like that rhythmic stepping in infants. So that highly driven like flexor extensor synergies.
Cool. Okay, so now more of a question about your plugin. This is from Dario Cazzola. Can you explain again how you identify the input synergy matrix W? It's great that you could input different matrices in order to couple muscles, but he was wondering whether you can input also experimental EMG data.
Yes, so you can make it whatever you want. And so, for example, in that prior study I shared with you today, we did just make random synergies. We have since made ones that are on EMG. Of course, the challenge with EMG is that your model often has more muscles than you have EMG channels. And so some of the strategies that we've used for that are either only specifying EMG or only specifying synergies, for those muscles that we have EMG data for, and then providing kind of an identity matrix for all the other actuators within the system. Or especially, you can also group muscles together, which also is a strategy that was used by Rick Neptune and Jessica Allen in their prior study. But so you have to be a little creative about matching what you have experimental data with what you have in your model. But there's several different ways you can test it out and try different things.
Great. So we have time for one or two more questions. The next is from Kenneth Smale. So synergies before the model are from excitations, while the synergies after are from activations. Might the model's activation or contraction dynamics account for some of the difference?
So this is static optimization, so there are not contraction dynamics implicitly modeled in it. I will say, though, that one kind of interesting part of this paper that I didn't present today, because it gets a little confusing and hairy, but I encourage you to go back to the paper if you're interested, is that there does seem to be an effect of motor variability. So if we go in and instead of just giving the exact same synergies for each force direction, if we add in a little bit of noise to the system, so for example, each different force direction that they generate with the hand, if I add a little bit of noise to my W matrix, my synergy matrix, and calculate the muscle activations, your ability to identify synergies does improve with that added kind of variability, which is another interesting component, and also one that supports that whenever you're calculating synergies or looking at synergies, whenever you average, for example, EMG data across a bunch of trials, you're getting rid of a lot of trial-to-trial variability, which actually is, important variability and can be useful in identifying those underlying synergies slightly more accurately.
Okay, so a somewhat related question from Yen Sun Wu. So what is your expectation about the results if the initial synergies are from EMG during the same or similar task?
Let's see. I'm not sure if I totally understand the question, but so if the synergies come from EMG, one of the challenges, again, with all of these is that we don't have EMG for all the models in the muscle. We've explored a little bit and also you have to remember that if you have synergies coming from EMG, you are already using one specific matrix factorization algorithm. So for example, you're using NNMS or one of those others to calculate your synergies. And so that algorithm will perform better at identifying synergies relative to the other algorithms. But we haven't looked extensively at the question of providing those EMG-specific synergies into the model.
Okay, so we'll take one more quick question and this is from Nicholas Bianco. So first, he says, great talk. And the question is, so why did you decide to minimize synergy activations versus muscle activations constrained by a set of synergies or some other cost function?
It was easier to implement in OpenSim is probably the quickest answer. So it was easier to give a set of weighting matrices and minimize those than to apply constraints and just the number of constraints that you'd have to apply. It was largely an OpenSim implementation choice. But you all can dive into the source code and try it the other way, and I will gladly test it.
Okay, great. Well, thank you, everyone, for all the great questions. Thank you, Kat, for a great talk. There are a few questions that we didn't get to, so we'll save those and can send those to you, Kat, and maybe you can try to follow up by email with the questions we didn't get to. So, yeah, can you bring up the last couple slides for me, Kat?
Okay, there's your questions. Very nice.
We took care of all the questions. So I do want to acknowledge our funding sources, which are on the next slide. So OpenSim and the webinar series are supported by several grants from the NIH, including the NCSRR, Symbios, and the Mobilize Center. We're also supported by DARPA through the Warrior Web effort. And then next slide. Thank you all for a really great discussion. Lots of really good questions. Thanks, Kat, for the really great talk and for sharing all those resources with the community. So if you want to find out more about OpenSim, upcoming events, and other resources, you can check out our website. And please complete the survey that will appear in a pop-up window at the conclusion of the webinar. We'll also send out a follow-up email that will have links to Kat's SimTK project and relevant publications. So thank you again, everyone, for participating. We hope you'll continue to stay tuned into the webinar series and stay involved with OpenSim. Thanks, everyone. Bye-bye.